Hello dear students, welcome to Biochemistry by Revati. I am Revati and in today's class we will continue the biological buffers and the physiological importance. If you want to know the basics of biological buffers and the bicarbonate buffer system, please watch the part 1 and part 2 videos. I'll give a link in the description below. Please go and check out. Now let's begin the class. Now we will learn one of the most important buffer systems in the body that helps maintain pH, that is phosphate buffer system. Now let's see where it works, how it operates and why it is vital for life. Phosphate buffer system, it is found in the cells and kidneys. What are the components of this buffer system? Dihydrogen, phosphate, that is H2PO4 minus and monohydrogen, phosphate, that is HPO4 2 minus. Now this phosphate buffer system works on this reversible reaction, that is H2PO4 minus dissociates to HPO4 minus 2 minus plus H plus. Now here H2PO4 means dihydrogen phosphate acts as weak acid. Weak acid means donates protons. And here HPO4 2 minus is monohydrogen phosphate. Now here this acts as weak base. Weak base means which accepts protons. Now let's see the mechanism. Now let's see how the mechanism works. The phosphate buffer system mainly works inside the cytoplasm of the cell. Why it works inside the cytoplasm of the cell? Because its pKa value is 7.2, which is very close to the physiological pH, that is 7.4. And also the concentration of dihydrogen phosphate and monohydrogen phosphate are ideal for buffering at this pH. Now what is this pKa? This we have already covered in the previous video. What is this? It is acid dissociation constant. Now, if you want to know the basics of this, I'll give a link in the description. Please go and check out. Okay. Now, let's see what will happen when the cell becomes too acidic. What will happen when cell becomes too acidic? Excess protons are released. If excess protons are released, means what will happen? Again, it leads to the instability of the cell. So, in order to neutralize this excess H plus ions, we are taking base form. What is the base form here? Monohydrogen phosphate. Now, this base form will neutralize excess H plus ions. Means here, monohydrogen phosphate combines with protons to form what? Dihydrogen phosphate. So what will be the result here? The pH does not drop too sharply. Keeping the cell stable. Now if you see in the second condition, when the cell becomes too basic or alkaline. Here if you take acid form, what is acid form? Dihydrogen phosphate. Now this dihydrogen phosphate will release protons or H plus ions or hydrogen ions in order to again neutralize the cell. So what will be the result here? The pH doesn't rise sharply, keeping the cell stable. Is it clear? Till now we have learned that the phosphate buffer mechanism inside the cytoplasm of the cell, and we have already seen that location is in the cells and even in the kidneys. So now let's see how it works inside the kidneys too. So here its role is to excrete excess acid from the blood and also to maintain blood pH. And let's see how the mechanism is involved. So here the phosphate buffer system mainly works inside the kidney tubules, especially in the distal convoluted tubules and collecting ducts. Now let's see the mechanism. So first here the kidneys will filter monohydrogen phosphate from the blood. So inside the tubules, means here this working mechanism is happening inside the tubules, right? So here inside the tubules, if the blood is too acidic, what will happen? So we have already seen in general, if it is acidic means excess H plus ions are released. So here also excess H plus ions are secreted into tubular cells, from there into urine. Now in order to neutralize this excess H plus ions, we have to take the base form, that is monohydrogen phosphate. Now this base form will combines with excess H plus ions to form dihydrogen phosphate, that is excreted into the urine, making the blood less acidic. So if you see in the second condition, that is if the blood is too basic or alkaline, what will happen? Reverse. Here the excess H plus ions are secreted. Here few H plus ions will be available. Now in order to balance the H plus ions, again we have to take acid form. What is acid form here? Dihydrogen phosphate. Now this dihydrogen phosphate associates to monohydrogen phosphate and hydrogen ions are released into the blood to balance. So here the condition will, the urine will be or blood will be less basic. So in both the conditions we have seen that the pH has been stable. So if it is too acidic, excess ions are released. If it is too basic, H plus ions are combined. Is it clear? Now let's simplify the phosphate buffer system with a small example, that is muscle cell. Now let's see, when you exercise, what will happen? Our cell produces a lot of acid, means during exercise. It will produce a lot of acid. Lot of acid means what again? Extra H plus ions. Now what phosphate buffer does? Now phosphate buffer does means it grabs extra H plus ions. So here the what is the component of phosphate buffer? That is monohydrogen phosphate. It grabs H plus ions and converts into dihydrogen phosphate. Here it is making the cell less acidic. Means it prevents the inside of the cell becoming less acidic. Now what will happen when the cell rest, muscle cell rest, environment become too basic. In that condition, what phosphate buffer does? It will release some hydrogen ions. So here these hydrogen ions will make the muscle cell less basic without rising the pH. Here without dropping the pH. So finally, what is the conclusion here? The phosphate buffer system maintains the pH 7.2 of the muscle cell and allowing the muscles to work properly. Is it clear? In kidneys, when the blood is too acidic, what will happen? The phosphate buffer system means the phosphate buffer grabs extra H plus ions, means extra acid. Here the phosphate buffer, that is base form monohydrogen phosphate, neutralizes extra H plus ions, means extra acid, to form dihydrogen phosphate, which is then excreted and eliminated from the body through urine, making the cell less acidic. Blood. And if the blood is too basic, dihydrogen phosphate releases some of the H plus ions or protons or hydrogen ions in order to make the blood less basic. So in both the conditions, phosphate buffer is maintaining the blood pH in the range of 7.4, acting as a second line of defense in the body, because we have the major or the first line of defense as the bicarbonate buffer system. You may get doubt that if bicarbonate buffer system is already available in the body, what is the need of the phosphate buffer system? Here, phosphate buffer system is also important because where the concentration is higher, especially in certain cells like kidney cells. In those cells, the phosphate buffer system may act its role. So, is it clear? That's all for today's video. I hope you have understood the concept clearly. If you have any doubts regarding the concept, please comment on your doubts. If you found this video helpful, please like, share and subscribe to Biochemistry by Revati for more simple and clear explanations. Until then, keep learning and stay curious. See you in the next class.